Hello viewers, welcome to this session on an overview of green biotechnologies. I introduce myself as Dr. Sushmita Bhaskar, Assistant Professor from the School of Interdisciplinary and Transdisciplinary Studies, Indira Gandhi National Open University, New Delhi. The greatest discovery on this earth, maybe in this century or in future, would be the discovery of life on another planet or on extraterrestrial space. That's because this earth, this planet that we have, is the only place where we have life, the presence of living organisms. But right now, human beings, especially anthropogenic pollution and human interventions, have actually destroyed our planet, and we are facing an innumerable number of contaminants, pollutants, and we are facing the environmental as well as the health effects. And in this lecture, we shall see the definition of green technology, how green technology could be used in the various sectors, the meaning of green biotechnology, and with special emphasis on how green biotechnology could be used in the food processing sector and in the food sector. So let us now get going with this lecture on green biotechnology. So now let us see the definition of green technology. As it says, it means it is an eco-friendly technology or it is environment friendly. So an eco-friendly technology that is used in a way which protects our environment and it also conserves our natural resources. So that is the actual definition of green technology. Now if we go on to some concepts regarding green technology, this refers to the products or the equipments or certain systems that can reduce or minimize environmental degradation. For example, reduce the amount of pollutants in the environment. It may be in the water ecosystem, it can be terrestrial on the land, or for controlling various purposes in order to reduce and minimize environmental damage. Again, it can produce zero and low greenhouse gases, for example, the use of biofuels or compressed natural gas, that is a CNG, which is being used these days presently. So all these can reduce and minimize the effects of the pollutants. Again, the green technology means it should be safe to all life forms, including the human beings. It conserves energy and natural resources, it promotes the use of the renewable resources, and it supports the use of natural organic resource. So that is what we mean by green technology. The different sectors where green technology could be applied, or their applications, this could be, as I said, in the energy sector, where we can use them in the form of biofuels for transport, again for the power production and the energy supply and consumption. In the construction buildings, here you could have green architecture and green infrastructure. So here again green technology plays a very important role. In the water and the waste management sector, where the waste can be suitably managed by using bioremediation or other green processes which can clean the systems. Again in the health sector, for the production of vaccines and drugs. So the novel compounds could be extracted from the plants or the alkaloids, and these could be also treated for the very severe or the health disorders and diseases, for example in the case of cancer or the Hodgkin's disease, lymphoma, etc. Again, this could also be applied in the food processing sector, where you can use the techniques such as biopreservation and in order to store and preserve the food products, also as the use of prebiotics or probiotics for enhancing the flavor of the food products in the food sector. So that was something about green technology. Now let us see some concepts and the definitions of green biotechnology. So green, again, as we have already mentioned, it refers to anything related to the environment, or it is an eco-friendly technology. And this green biotechnology involves the using of genetically altered plants or animals for eco-friendly farming solutions, or as an alternative to traditional agriculture, horticulture and the animal breeding processes. Now this can tackle the food security issues. It can also manage environmental pollution from the various sources. It could be point or non-point sources, and it can also explore plants as biofuels. Going on to the applications of the green biotechnology in the food sector. So we have the food biotechnology. In this, you can use the techniques and processes using living organisms or substances in order to improve the quality of the food. So this could be like in the form of probiotics or prebiotics. They are also known as the starter cultures, and they ensure food safety, and they can also enhance the food value of a substance, which can be also further certified. The second application is the use of green biotechnology in agriculture, is also referred to as agricultural biotechnology. Here the plants or the living organisms can be used for the crop improvement, and genetic engineering can also be used to protect the crops against certain pests, certain infestations. So, this could be in the form of biopesticides or biofertilizers. Here in this figure you can see organic farming practices being practiced by the farmers in a village in Kerala, and here there is zero pollution, and the farmers have managed the paddy fields really well by the use of organic farming techniques. The third application would be in the field of environmental biotechnology. So, here the living organisms could be used to clean up, it could be used to clean up the pollutants from the various industrial sectors, and also the waste waters, also solid waste, different types of the pollutants could be cleaned up, and even some plants and animals have tolerance to heavy metal. They can even sequester the heavy metals onto their cell walls. Certain plants, their roots also, they can absorb the heavy metals and they can sequester the same. So, in this way, in a very green and eco-friendly way, we can monitor and protect and treat the pollutants in a eco-friendly way. So, these are some examples, and here again we find a place at Thailand, that is at Kanchanaburi. It is a wetland where the organic practices are being followed, and you do not see a sign of the chemical or the pollutants, and the people living there are quite healthy. Coming on to the use of green technology in the health sector, here again vaccines and drugs could be produced from using this technique. And for example, in the past 20 years, we have seen the evolution of highly infectious diseases. So, even when we go to the doctor for a treatment for a fever, sometimes they are not able to be diagnosed well. There are different forms of viruses. For example, we have the Ebola virus, then we have seen this avian flu, then different forms of influenza viruses. So, all these are creating panic among the people in the living in, especially in the populated areas. So, we have seen a rise of infectious diseases which are very difficult to treat, and these diseases account for 30% of the global death. So, maybe the green biotechnology could help in inexpensive diagnostic procedures and novel and quick methods of drug delivery. Powerful nanotechnology solutions also are coming up that can repair the DNA damage, and they can also protect the cellular damage. So, this technology has also been very helpful in the pharmaceutical industry, where the pharmacists are able to produce the drugs. So, earlier we had the chemical synthesis of drugs, but now there are alternate ways where you can take out the very novel drugs. For example, CSIR has come up with the BG34 and other IME9, all this which are used to treat the sugar and the diabetes patients. So, these novel techniques can be used in order to extract the efficient proteins, or the components that could be used to treat infectious diseases. So, now that we have an overview of the different sectors where green biotechnology can be used, the emphasis of this lecture was more on the food processing and the food sector, the food industry. Let us now go on to the green biotech in food industry. So, food is a very essential component, we all know, and every morning and every day we need all the essential elements, the amino acids, the vitamins, carbohydrates, the lipids, proteins, all in the balanced proportion to make us going, and so that we are on the move. So, the population growth is also one factor. We have a billion population, especially in India and China, the most populous countries. Because of the populous growth, we have the need for sustainable food production and processing technologies. So, maybe green biotechnology could be a solution in order to address these problems and challenges. And these technologies could also reduce the amount of toxins that are released, because certain processes, chemical processes, can release toxins into the food. So, maybe this technology would give you an added benefit of reducing the same. And also sustainable diets. People are now moving on to sustainable diets globally, and so the biodiversity is also being surveyed for the genetically superior varieties of rice, which the populations, for example, in the belts of Chhattisgarh and Madhya Pradesh, people still practice the farming practices where we find genetically superior variety, and the protein content is also very high in these type of the rice. So like this, genetic engineering and the biotechnological approaches could help in solving the crisis of the food production, and in order to meet the sustainable food production. So what could be the advantages of this in the food sector? The quality, safety and nutritional properties could be enhanced. Then biopreservation techniques, these again they give you longer shelf life for a product. Electromagnetic wave heating, electric magnetic fields, non-thermal technologies, all this can reduce the process related toxins in the food and also the environmental impact of the food, that is packaging. So this also plays a very important role in liberating the toxins, and also sometimes pathogenic bacteria can also liberate the metabolites, which can also cause problems of food spoilage. So maybe this could be one technology which could help protect our food and the food products. Giving an account of food-borne diseases. So if we take the USA, acute gastroenteritis affects more than 250 million people annually, and approximately 30% are due to the foodborne infections, that is the foodborne diseases. So this mainly the sources come from meat, from poultry, from egg, maybe from fishes and the dairy products. So all these may not be cooked well, or the organisms can easily attack. It depends upon the type of microbe. It could be thermophilic or it could be psychrophilic, mesophilic. So the organisms that live at the different temperatures, they can attack the food products and cause disease. The bacterial pathogens that are causing them, they could be, for example, Salmonella, Campylobacter, E. coli, Listeria monocytogenes, Staphylococcus aureus and Clostridium botulinum. So these are extremely pathogenic organisms. They release toxins, for example, in the botulism. So they create and they produce the toxins in the body. And there are certain methods like the physical methods where high temperature is used, and also chemical methods where the chemicals are used in order to preserve the food and to protect them from these organisms, but they may not be so protective in nature. So do we have solutions for the same? Yes. So green biotechnology does play a role in the control of spoilage of these foods. Innovative technologies can be used for controlling the microbial spoilage and safety hazards in foods, and bacterial antimicrobial systems such as the lactic acid bacteria, in short we say it as the LAB, and or their bacteriocins, that is what they produce. So these could be effective solutions in order to protect us from these pathogens. So biopreservation is one such innovative methodology. In biopreservation, the shelf life could be extended, enhance safety of the foods by using natural controlled microbiota and or the antimicrobial compounds, and it is an ecological approach to the problem of food preservation. Fermentation, this is traditionally been used for a long time. It's a common form of the biopreservation, and this is based on the growth of microorganisms in the different food, for example, in making yogurt or in the brewery, in the distillery. So they can be added as the starter cultures also to enhance the flavor of a certain type of alcohol or beverage. So these microbes are lactic acid bacteria that produce organic acids. In addition, they also have antimicrobial properties. As already been said, they add the value of the texture and the taste to a specific kind of food or beverage, and traditionally this has been used for a number of years, and the food has been preserved in such a way, in a bio and eco-friendly way. Presently, if we take into account, the fermented foods are on the rise, and they are popular even in the industrialized and the developed world, and 60% of the diet in the developed world comprises of the fermented food. So the fermented bamboo shoots. So recently I had been to the northeast of India, and there I found this fermented bamboo shoots where people prefer to have them. So these are really rich in the protein content and good for the body. So, this assures homogeneity, quality and safety of the products by the intentional use as the starter and protective cultures. Now, in this diagram if you see in the mixed and undefined starters, and when we use a traditional fermentation technique, then maybe the safety and the shelf life is not so long. And if we use the defined starters with proven industrial properties, then the reliability and the taste is good and there is also consumer acceptance. Now again if we have the defined strains with proven health benefits, so that is why we are adding these prebiotic and probiotic type of the yogurt that we find in the market or even ice creams. So they have they are very good for the health and they are good in protein, it is good for health and wellness, and also it is certified. So there is advanced consumer acceptance for such kind of food. So green biotechnology has played a very important role in the food industry and the food protection and safety. So what do we mean by starter cultures? So anything, it is like to start the engine or to start a conversation, so something which begins or starts. So starter cultures are the preparations of one or more type of microbes and they are used for initiating this fermentation of food production. They can be used in the dairy industry, in the meat industry, in the juice and also in certain other vegetable products. Some examples include the lactic acid bacteria. Among this also we find the Lactococcus, Streptococcus, Lactobacillus, Pediococcus, Leuconostoc, Enterococcus, Cornobacterium, Aerococcus, Enococcus, Tetragenococcus, Avagococcus and Weissella. They are gram positive, non-motile, non-spore forming and they ferment the carbohydrates to form lactic acid. Going on to the characteristic of the lactic acid bacteria, they have low amounts of guanine and cytosine in their DNA, that is less than 55%. They are resistant to bacteriophages. They have high resistance to freezing and lyophilization. They are capable of adhering to the digestive mucosa and they produce antimicrobial substances. So, a host of beneficial aspects and advantages from these group of bacteria. Now, again advantages of LAB. They produce number of bioactive metabolites during fermentation. They involve bacteriocins, biogenic amines, exopolysaccharides, proteolytically released peptides and so on. The second is prebiotics. These are added to food fermentations to improve the performance of probiotics. And research is still on in the fermentation of prebiotics such as fructooligosaccharides, which can help prevent osteoporosis, cancer, that is colorectal cancer and also obesity. So these are some of the beneficial aspects of the lactobacillus or the LOB group of microorganisms. The LAB bacteriocins, the antimicrobial ribosomically synthesized peptides, these are produced by the members of the lactobacteria and they are called as the bacteriocins. Such peptides can kill the neighboring cells, that is the pathogenic bacteria, or the peptides can kill the closely related microorganisms, and due to their nature they are inactivated by proteases in our gastrointestinal gut. So these are some advantages of the bacteriocins. So do we have some more benefits of using this LAB bacteriocins in the food preservation? Yes, we do. The characteristics are the protein nature, inactivation by the proteolytic enzymes of the gastrointestinal tract. They are non-toxic to the tested laboratory animals and they are genetically non-immunogenic. They are also inactive against the eukaryotic cells. They are also thermoresistant. They maintain antimicrobial activity even after pasteurization and sterilization. They have a broad bactericidal activity. And another useful feature is the genetic determinants of these are located in the plasmid, which are useful in the genetic manipulation in order to increase the variety of natural peptide analogues with desirable characteristics. So they can be used in the dairy products, nisin and nisin producing strains. They are commonly used against the pathogenic bacteria like Clostridium botulinum in a variety of cheese and Listeria in camembert and ricotta and manchego cheese. So they are used in variety of cheese in order to protect from the pathogenic bacteria. Some more varieties of the bacteriocin are the pediocin in milk, cheddar cheese, Munster cheese against Listeria, Staphylococcus aureus and E. coli, lacticin which is used against Bacillus cereus in cheddar and cottage cheese and yogurt, enterosin AS48 against Bacillus cereus, Staphylococcus aureus and Listeria in milk and Manchego cheese. So, these are some strains of bacteria which are useful in protecting us from the pathogenic from the pathogens or the pathogenic microorganisms in cheese and in other dairy products. In the same way, these bacteriocins can also be used for the biopreservation in meat and also the fish products, in also the poultry products. So, in the case of meat are actually complex systems and there are a number of growth factors that influence the microbial growth and also metabolite production. So, here we have seen nisin, enterosin AS48, enterosins A, B, leucosin, pediosin, all this either it could be used alone or in combination with several physicochemical treatments, and they can control some pathogenic microorganisms and also is found to be effective against Listeria monocytogenes. The biopreservation in vegetable foods, that is in the tinned food or the canned food. So, this is also common among a wide variety of people. So, here lot of organisms could enter the canned food. So, nisin is used effectively in the tinned vegetables and in fruit juice. Pediosin PA 1 by ACH in salad and fruit juice. Enterosins also have been found to be useful against Bacillus cereus in rice and in vegetables. So, these are some proven research that has been done on these bacteriocins which have been found to be useful in the vegetable canning and the tinning sectors. In the same way, biopreservation of fish is also important. It could be in the raw fish or in the frozen and the thawed fish. So, here deterioration of fresh fish, normally we have number of factors, but the gram negative bacteria are more responsible for this. We have the vacuum packed fresh fish, that is a seafood which have a host of pathogens that include the Clostridium species, Listeria species. Again, the combination of nisin and microguard, this has been found to be very useful against the aerobic bacteria, and the fresh chilled salmon here again, there it is found to have increased shelf life of these fishes, and it reduces the growth of the Listeria in the frozen thawed salmon. Now, coming on in continuation with this, there is another technology known as a hurdle technology. This was introduced by Leisner in 1978, and this states that the microbial safety, stability, the nutritional qualities of food are based on the application of several combined preservative factors and that the microorganisms are able to overcome. So, this hurdle technology refers to that rather than giving a single preservation treatment, we can go for a combination of various physico, chemical and biopreservation treatments in order to inhibit the microbial growth. In relation to this, a novel concept of multi-target food preservation has emerged in relation to the hurdle technology, and this gives a more effective preservation of food by the application of multiple soft treatments like that would disturb the homeostasis or the metabolic balance, metabolic exhaustion. It would avoid stress reactions by bacteria, and it is more effective to employ several small intensity preservation factors rather than using one single large intensity factor. So, this would be more beneficial and it can produce a synergistic effect. In this lecture, we have understood the definition of green technology, green biotechnology, and where the green technology as well as biotechnology could be used. We have also given a special emphasis on the green biotechnology that could be used in the food industry and in the food sector. For example, the various biopreservatives that can be used for preventing pathogenic organisms and preventing the health of human beings. Thank you for your patient listening.